So we just talked a little bit about how some of the metabolic pathways of carbohydrate, lipid, and protein metabolism all kind of come together. And now I want to talk briefly about the regulation of this and cell signaling. In terms of regulation and cell signaling, I think we want to kind of remember essentially our bodies are always either in an anabolic state or a catabolic state. So we really can't, within a given cell, it can't be going both directions. So essentially, either the cell senses that the body needs energy, in which case it's going to move down the metabolic pathway, if you want to think about it, the one we just looked at. Or if we don't need energy, then it's going to move into that anabolic state where it's building up tissue if it has energy there, for example, storing fat and that sort of thing. So kind of remembering that we have to, somehow our body has to be able to regulate and determine, do we need to make energy or do we need to store energy if there's energy to be had? And then if we need energy and it's not coming in, where are we going to get that from? So really we're either in an anabolic or a catabolic state for the most part. So then our body needs to have a way to determine whether we're moving towards catabolism or anabolism. So essentially, do we need to store energy or do we need to use energy and either use the energy that's coming in through a food source or break down energy stores such as fat stores or that sort of thing, or glycogen or whatever. So there's some key regulators in the body. There's a variety of regulators, but a couple that are really key that we'll focus on. So one is AMP, activated protein kinase, or AMPK, and another is malonyl-CoA. So we'll look at these just a little bit. First of all, with AMPK, I just kind of want to point out that protein kinases are a large family of proteins. They're enzymes that function really as key metabolic regulators in the cell. And they function by phosphorylating proteins, which then changes the protein function. So we remember if there's a different structure, a different group, like a phosphate group on a protein, it's going to change the function. So AMPK is a protein kinase that is activated by AMP levels. So for example, when we have a high AMP to ATP ratio, that signals that our body has low energy stores. So that's going to trigger more of that catabolic process where we're either going to use what's available for an energy source or we're going to break down something like glycogen or fat stores. That AMPK then stimulates a variety of reactions that allows this to happen. So for example, AMPK would stimulate GLUT4 translocation. We talked about GLUT4 in a previous unit when we talked about carbohydrates as the molecule, the protein that transports glucose into the cell. It's also stimulated by insulin. So a variety of these cell signaling molecules that cause all these reactions to happen. AMPK then can stimulate glycolysis. It inhibits fatty acid uptake, which then is going to cause beta oxidation of fatty acids, so we can break down those fatty acids. It inhibits protein synthesis. So just kind of a wide range of activities that once this AMPK is stimulated, it triggers all these other metabolic aspects. And again, looking at all three, protein metabolism, lipid metabolism, and carbohydrate metabolism, it affects all of those pathways. So that's one example. And then we'll look at malonyl-CoA as well. We can look at this pathway again for another regulatory step or regulatory compound, I guess, in malonyl-CoA. So for example, if the ATP to ADP ratio is high, meaning that there's a lot of ATP in relation to ADP, that means there's adequate energy and we need to store some of that energy. We don't need it as much. And so in that case, remember that acetyl-CoA, the tool here, our acetyl-CoA here really is a key step in the metabolic process. Remember, we kind of talked about right in here being really key. So if our energy needs we have adequate energy, so we need to store some of that acetyl-CoA can be transferred back to the cytosol, because remember, these pathways, this energy production and the TCA cycle happens in the mitochondria. So that acetyl-CoA can be transferred back into the cytosol and can be carboxylated to form that malonyl-CoA that we just looked at. So if I'm just going to little kind of abbreviate malonyl CoA here, and that is really comes into the metabolic process right here. So it's the first step in that fatty acid synthesis from acetyl CoA to fatty acids. Malonyl CoA is a product right here. So essentially, when we don't need energy, that carboxylation of acetyl CoA happens and triggers production of malonyl CoA, which then allows fatty acids to be produced. So just another example of a regulatory pathway that essentially, when there's more of a particular compound present, it stimulates the reaction to go one way or the other. When we think about all these metabolic pathways, I think it's also helpful just to keep kind of in the back of your mind where all this is happening. So over here we have our little guy that's the whole, oopsie, the whole human body there, our whole little guy. And then within, inside there's different organs. So, we have all these different organs, the intestines, we have muscle tissue down here, all of those kinds of things. So within each of those tissue types or organs, we have cells, obviously. And then within the cells, we have different parts of the cells. So we have our cell membrane that's really important for some reactions. We have our little mitochondria here that aren't labeled but are there, the cytoplasm where some of those reactions happen. So I just think it's helpful to kind of keep it in context and think about, like we just talked about, how the TCA cycle or the Krebs cycle happens in the mitochondria, whereas other reactions happen in the cytoplasm. So things are moved to different places in the cell based on those regulatory factors. The other thing to consider is that different cell types function a little bit differently. Again, remembering back to, for example, carbohydrates and those glucose transporters, that some types of glucose transporters are active in certain cells versus others. For example, we talked about the GLUT4 being very active in muscle tissue and adipose tissue. So depending on the cell type for what organ it is or where it is in the body, there may be different molecules that are more or less active. When we talk about different organs and cells in different organs having different functionality, it's really important to always think about the liver when we think about metabolism. So the liver is really central to metabolism. It's a key processor and distributor of carbohydrates, proteins, and lipids. So it handles all of those. So the liver can in the liver, glycogenesis as well as glycogenolysis happen. So basically glycogen can be built up or broken down. So remember, the liver is one place where glycogen is stored and the muscle is the other place. So that's really our only store of carbohydrates, and it's a relatively small store. But in the liver, that glycogen can be broken down when glucose is needed, or it can be made when there's excess glucose available, for storage. The liver is where proteins are synthesized. There are also proteins are broken down. They're converted to other compounds. So remember, proteins, the amino acids within those proteins are really important for making different compounds. In terms of protein, the liver is also the only organ or cell type that can form urea. So it takes that nitrogen from the protein compounds and turns it into urea, which then can be excreted from the body so that we don't build up toxic levels of ammonia in our blood. We've talked about, when we talked about lipids, the lipoproteins that are the lipid protein compounds that transport lipids throughout the body. So that protein portion of it allows the lipid, which is not water soluble, to be transported through the blood to the tissues where they're needed. So in the liver is where all of that synthesis of the lipoproteins happens. So the LDL and the VLDL and the chylomicrons and all of the packaging and repackaging to move things throughout the system. So the liver is a huge component of metabolism, and it's essential that we have adequate liver function and healthy liver function in order to metabolize nutrients properly.